Hi, this recording, okay, or this video is for those taking the 5017 Combined Science Biology G2. So if you are G3 Combined Science or G3 Pure Science, please ensure that you save this, because tomorrow is the bio exam. Okay, because tomorrow is the bio exam, right? I have nothing much to cover. Okay, it's really relying on your memory as well as your content. Okay, so today I'm going to cover a total of 5 chapters, but I'll run through very fast because time is running short. So let's start. First up, you need to tell me what is an animal cell. Okay, like you need to be able to state the function of each organelle for the animal cell. So you need to tell me like cell membrane. Okay, what's the function? The function is a partially permeable membrane. You must tell me that it surrounds the cytoplasm of the cell. You must also state that it controls the movement of substances in and out of the cell. Okay, in and out of the cell controls movement of substances, not just water. Okay, a lot of people, they are tempted to write water, and it's actually wrong. Okay, for cytoplasm, I don't need you to memorize gel-like matrix. I just need you to memorize site of various chemical reactions. And for nucleus, I need you to memorize controls all the activities of a cell, growth, repair, and cell division, as well as it contains chromosomes, which are made up of DNA. Okay, the next one is okay, this endoplasmic reticulum and the Golgi apparatus, please don't memorize. Okay, that one is because of the notes, they are meant for both G3 and G2, but today I'm going to cover what G2 has. Alright, so you also need to state the function of mitochondria well. So you need to tell me that they are small elongated organelle, and aerobic respiration will occur when the where energy in the form of the aerobic respiration is extracted from food substances in the presence of oxygen. Okay, you also must state that the vacuoles are small, and vacuoles, the function is they are small fluid filled vesicle bound by a membrane that stores water and food. You can also say water and nutrients, is fine. Alright, then for the next one, plant cell, you need to be able to state 2 more organelles, mainly the chloroplast and the cell wall. What are the functions? Okay, chloroplast, basically when you draw, it must be oval membrane bound organelles filled with chlorophyll, and they are the sites of photosynthesis, which is the process by which plants actually make food. Right, for cell wall, you need to tell me they are made of cellulose, and they actually are the supporting pillar, or you can say it provides support to maintain the shape of the cell. Alright, okay. For chapter 3, you must also state, for point 3, you need to state the adaptation of red blood cell. So you need to tell me that red blood cell, they contain hemoglobin, it does not have a nucleus, and it has a biconcave shape. Okay, hemoglobin, you need to tell me it's an oxygen carrying protein. For no nucleus, you need to tell me that it has more space for hemoglobin to carry more. Alright, it has more space to allow hemoglobin to carry more oxygen, and also you need to tell me that it has a flattened biconcave shape, right, which enables them to have a higher surface area to volume ratio. Alright, for what? For faster diffusion of oxygen. Alright, therefore the xylem vessel, you need to tell me that the xylem vessel are elongated hollow tubes. They are dead at maturity, and the adaptations to this function include the following. Alright, the adaptation you don't really need to know, you just need to know that they are elongated hollow tube, as well as they are dead at maturity, and that they conduct water and mineral salts from the roots to the leaves of the plant. Okay, can? Alright, please pause the video if you need to revise or you need to read the content, yeah, because I'm going quite fast today. Okay, for C, right, you need to tell me what are the adaptation of root hair cell. So root hair cell, you need to state that they are elongated non-narrow structure called the root hair, as well as increasing surface area to volume ratio of the cell, which result in faster absorption of water and mineral salt. Okay, You also need to tell me what is the organization of multicellular organism. You tell me that the cell is a basic unit of life and each cell has a specific function. Tissue is a group of cells. Organ is a group of tissues with similar to perform similar function. Organ system is a group of organs, and organism is a group of system. Okay, organism can be your animals, your plants, can be anything, right? So long as it has a group of system. Right, that is it for chapter 1. Okay, for chapter 2 is movement of substances. So first one I want to cover is diffusion. Okay, diffusion, you need to tell me there is the net movement of molecules from a region of higher concentration to a region of lower concentration down a concentration gradient. But you need to add one more thing here, until equilibrium is reached. If you don't know how to spell equilibrium, please Google it. Alright, and you tell me what's a concentration gradient. Is basically the difference in concentration between a region of higher concentration of a substance and a region of lower concentration of the substance. Okay, and when the gradient is steeper, the rate of diffusion will be faster, right? And when it exists, diffusion will take place until particles are evenly distributed throughout the region. Okay, you also need to tell me that the cell surface membrane is a partially permeable membrane, okay, that allows gas such as oxygen and carbon dioxide to pass through. Okay, in the example for diffusion in animals, you don't really need to know. You need to know about active transport. Okay, basically you need to tell me that is the process which energy is used to transport substances, alright, across a membrane against a concentration gradient. Okay, example can be like your root hair cell, for example. Alright, and the energy used for active transport is obtained, that means it comes from cellular respiration. You need to state that as well. Okay, point 3, osmosis. You need to tell me that osmosis is the net movement of water molecules from region of higher water potential to region of lower water potential through a partially permeable membrane. You need to state partially permeable membrane. That three words are super important, okay, in the osmosis definition. Okay, and you need to tell me that how they flow. Alright, from dilute to concentrated, how does it flow? Alright, which one has a higher water potential and which one has a lower water potential, and state the reason. Okay, so an example is like now here about the beaker, about dilute solution and concentrated solution. Okay, for the example, you don't really need to know, except for this. You need to tell me that the something, okay, be it a solution, has a higher concentration of a blank, okay, or something, than the 10% something solution. Okay, hence it has a lower water potential compared to what sucrose solution. Okay, and for osmosis in plant cells, you need to tell me that the vacuole increases in size, and the expanded cell contents exert pressure on cell wall. This is for higher water potential, yeah. And you need to tell me that the cell wall exerts an opposing pressure on the cell contents, preventing the entry of more water, and it prevents the cell from overexpanding and bursting. And at that point, the plant cell is already very turgid. Okay, you need to learn the word turgid. Alright, and the pressure provides mechanical support for many non-wooden plants. That toggle pressure you don't really need to know. Okay, the next one is the lower water potential. When a plant cell is immersed in a solution with a lower water potential relative to its cell set, the water diffuses out of the cell into solution by osmosis. You need to tell me osmosis, super important keyword. Right, and because of that, the vacuole will shrink, and then the cell will stop exerting pressure. Therefore, the cell will become flaccid. Don't use limp. Okay, flaccid. Flaccid is the keyword. And if more water leaves the cell, it will shrink until to an extent that the cell surface membrane will pull away from the cell wall, and you tell me the process is called plasmolysis. Okay, plasmolysis. Yeah. Okay, next one, animal cell. You tell me that it's the same thing as plant cell, except for this time around you use the word as cytosis. Okay, when the animal cell swell and burst. And then for low water potential, you need to tell me that that in red blood cell, the little spike appear on the cell surface membrane. It will known as crenation. Okay, crenation with an N, not an M. Okay, not an M, but an N. Okay, got it? Right, that's it for chapter 2. Moving on to chapter 3 will be biological molecule, as well as chapter 4, which is enzyme. Okay, you need to know this. What are carbohydrate? You need to tell me the carbohydrate, what it stands for. All these are given in the Gmail that I've sent you. Okay, so you need to go and take a look. Alright, sorry, not Gmail. This thing will be here for your reference. Alright, now you need to tell me what are simple carbohydrate, what they contain, as well as the three main group. Now the three main group don't really need to memorize. Okay, but I need to state the example. So like monosaccharide, you need to tell me there is glucose, fructose, and galactose. And for disaccharide, you need to know that because di means two, so you tell me it's formed by maltose, sucrose, and lactose. Okay, I think they are fairly simple. For complex, they are called polysaccharide. Okay, which you need to know the example of starch, glycogen, and cellulose. Okay, these are some of the examples. Alright, and over here, this one. Okay, that's for reducing sugar. Now for reducing sugar, it only applies when you have glucose, fructose, galactose, maltose, lactose. Okay, sucrose is, take note, sucrose is never a reducing sugar. Sorry, sucrose, I repeat, sucrose is never a reducing sugar. Okay, you need to also tell me that the test for reducing sugar is a Benedict test. Okay, and you need to know the procedure if you need to. But the color is the super important one. Okay, it will change from green is traceable, to orange, which is moderate, and brick red, which is high. Okay, it depends on the increasing amount of reducing sugar present. Okay, next one is test for starch. This one will be for your iodine test, and you need to know that if the sample is a positive result, it will turn blue black. Okay, from something to blue black. Okay, but blue black very important. Okay, next one I want to cover is fats. Yeah, you need to know that it's made of carbon, hydrogen, oxygen, and they are of two types of smaller molecule called glycerol and fatty acid. Okay, this is a fat molecule example, and then there are storage molecule. Okay, how do you test for fat? For fat, right, you need to know that it's an ethanol emulsion test. Alright, and an example. So you need to tell me that if the sample has a positive result of fats, a white emulsion will be formed. But I do not like to use the word white emulsion. Please use the word, if fats are present, a cloudy white emulsion will be observed. Alright, and the last one is protein. Okay, they are made out of carbon, hydrogen, oxygen, nitrogen. They may, in some cases, in very rare cases in humans, can also contain sulfur. Alright, and proteins are made of amino acid. Okay, you should know what an amino acid is. Okay, it was covered in your Set 2 science. So you need to know that amino acid link up in a condensation reaction to form a polypeptide chain. You need to use the word polypeptide, super important. Okay, polypeptide is the important keyword. Alright. Okay, this is what a protein molecule looks like. You need to memorize because the question will give you. But you do need to know what is denaturation. So denaturation is the protein will be denatured if they are heated or placed in an environment with unsuitable pH, and that the denaturation usually leads to loss of function as proteins require their 3D shape to function. Okay, and the last test for protein, okay, you need to tell me it's a Buret test, and if the proteins if the sample has a positive result of protein, you need to tell me a violet color will be observed. Alright, negative will remain blue. Okay, chapter 4, enzyme. You need to tell me what an enzyme is. So an enzyme is basically biological catalyst. Catalyst, important word. They are made of protein. Okay, enzymes are biological catalysts. This is a definition, by the way. Okay, make sure you know this thing well. Alright, they are substrate specific. You need to tell me that they are substrate specific. Okay, and then the substrates are reactors that enzyme acts on. Each enzyme can only act on a particular substrate of the reaction they are supposed to catalyze. Okay, this is for enzyme definition. Next one is this one, super important. This is the most important element of the whole enzymes chapter, is your lock and key hypothesis. You need to know what is the lock and key hypothesis. So the lock and key hypothesis, what does it mean? It means that it relates to enzyme specificity to the presence of active site. Okay, you need to tell me what's the active site. Okay, it's basically the region that the substrate binds to, and it's usually the pocket or groove on the surface of the enzyme, part of the enzyme unique 3-dimensional complementary structure. Okay, they are missing one word there, it's complementary. Alright, and when they are bind together to the active site of the enzyme, they will form an enzyme substrate complex. Alright, and then it will be catalyzed. Okay, then there's a effect on temperature. This one I will just scroll a bit slower so that you can take a screenshot if you need to. Okay, so this is for enzyme. You also need to tell me that the enzyme, okay, once it reaches high temperature, the enzyme is completely denatured and the rate of the reaction will drop to 0. Okay, it's straight 0. And also you need to tell me that at the highest point the enzyme is of optimum pH. Okay, especially for this part C over here. Okay, if you can see the graph here, it says effect of pH on the rate of enzyme of amylase. You need to tell me that this point over here is the optimum pH. Okay, optimum pH. Okay, that is it for chapter 3 and 4. Moving on to chapter 5. Okay, you need to tell me what are the processes of the digestion. Okay, you tell me what the mouth does. Chewing of food by teeth, physical digestion. Okay, you need to know the chemical name. That you need to know that chemical digestion start in the mouth as this one, salivary amylase. This is an enzyme. It breaks down starch into maltose. You need to tell me those two things, okay? They are important. At this point, right? Okay, this one you don't need to know. This one is just about the bolus, which is not covered in any syllabus. Okay, for the esophagus, you need to tell me that the food reaches the stomach after passing through the esophagus. Okay. Yeah. Okay. Can. Alright. The next one is the stomach. You need to state what are the gastric juices. Okay. They are hydrochloric acid as well as pepsin, and they work and they provide an acidic environment about a pH of 2 for pepsin to work well, and it kills bacteria in the food. Okay, the chemical digestion of protein starts here as pepsin digests protein. Alright. And then for small intestine, you need to know the three parts: duodenum, jejunum, and ileum. Okay, and you also need to know about in the small intestine about the pancreatic juice. It's also very important. Alright. And before fats are digested chemically by lipase, this is also important. Bile acts on it first through the process of emulsification. Okay, basically what happens is the fats, okay, or I'll say the bile emulsifies into fats. Okay, you need to know that. Okay, I repeat again, biles need to emulsify to be able to become fats. Okay, these are all the digestive enzyme, but the more important one I would like to highlight is this one. Must mention polypeptide chain. Okay, you do not need to know trypsin because trypsin is not covered. Peptidase also don't need. Lactase also don't need. Okay, so all the things you need to know is amylase, pepsin, maltase, and pancreatic lipase. That's it. Okay, only 4. Okay, you need to tell me what's assimilation also. The absorption of digested food, which is mainly the jejunum, but they rarely test. By in the event that they test, you need to know the definition. Alright. Okay, and moving on down to the large intestine. Sorry, for the small intestine, you also need to state this. Glucose and amino acid are absorbed by diffusion or active transport depending on the concentration gradient. You need to know that as well. Alright. And the last one is the large intestine. What does it do? Okay, it comprises mainly cellulose, indigestible to human. Okay, elimination is called egestion. You need to know egestion. Okay, how do I remember? Egestion, eject. Okay, like how you eject, you need to tell me there is egestion. Alright, and the role of other organ, there's a liver, the gallbladder, and the pancreas. You need to remember, but I think the pancreas and the liver are the more important ones. Okay, transport of product of digestion. Okay, you need to know that the largest blood cell is called the hepatic portal vein, which leads to liver. Okay, why? Because the hepatic portal vein, okay, the largest one, connects to the liver such that there will be enough blood flow. Alright, to keep your liver healthy also. Alright. Alright, you need to say that the blood from small intestine travels to liver via hepatic portal vein. Alright, that is the reason is because the hepatic portal vein is a shortcut to the liver. Alright. Okay, the next one are roles of liver. This is super important. You all of you must know what are the important roles of liver. Okay, when the blood glucose concentration is high, okay, insulin will be secreted, okay, from the cells of the insert of Langerhans. Okay, you need to tell me is an insert, the islet of Langerhans. Alright, and you need to know the effects of it. What does it do? It converts excess glucose into glycogen. You need to know that. Okay. But if blood glucose concentration is low, the glucagon will be secreted. Okay, how do I know? Low glucose equals glucagon, high glucose equals insulin. Okay, I need to know is from the cells, again cells instead of Langerhans. Alright, but the thing that's different is the effect. The effect, it convert glycogen into glucose. So whatever happens in the high blood glucose concentration will be the opposite for the low blood glucose concentration. You also need to know the liver produces bile and stuff. Yeah, I think this one more important. Hemoglobin from red blood cell is brought to liver where it's broken down. Yeah, that's more important. Then you need to know what is deamination of excess amino acid. Okay, you need to know what is deamination. Yeah, what is the function of deamination? What is the definition of deamination? You need to know. Okay, it refers to the removal of the amino group N minus NH2 from amino acid. Okay, and the remnant will be converted to glucose. Okay, starch. Okay, you also need to know what is detoxification. Basically, it's like break down alcohol. So if you drink alcohol, the liver will help to detoxify it. Okay, yeah, that's all for this is chapter 5 if I'm not wrong. Yeah. Okay, chapter 6. Wow, finally the last one. Okay, transport in human. You need to know the 3 parts covered in secondary 1 and 2, also a bit of your primary school science. Okay, the circulatory system you need to know is heart, blood vessels and blood, and the circulation method. Okay, systematic and pulmonary. So systematic is like carry rich oxygen, whereas deoxygenated blood is poor oxygen. Okay, so the systematic circulation is from the heart to all body organ, whereas for the deoxygenated, the poor one will be from the heart to the lung for gaseous exchange before they return the blood back to the heart. Okay, yeah, so this is what the diagram looks like for single and double. Okay, I think double is easier to memorize because single, right? Single, you will need to know what this arrow stands for. You need to tell me there's artery. Okay, then this one it should be vein, which leads me to this one, main vessel of human circulatory system. Okay, now the main 3 types that you learn until sec 2 is artery, vein and capillary, but this time you have 5 more to learn. Okay, you have 5 more to learn. You need to know. The first one is pulmonary artery. It carries blood from the heart to the lungs. Okay, because pulmonary, that means it react from the opposite. Okay, it's opposite, yeah. Alright, opposite from the actual function. So you need to know that the function of artery is carry blood away, right? But pulmonary artery, because it involves the lung, you need to tell me about the lung. Okay, and it also locate on the right side of the heart, contain more carbon dioxide. Okay, how do I know which is okay, later I'll cover what is left, right, atrium and ventricle, yeah, as well as the valve. Okay, because that one you also need to know. For a pulmonary vein, you need to know that it carries blood from the lung to the heart. It's located on the left side and it contains more oxygen. Okay, for artery, pulmonary artery, it contains more CO2 because you need to go back to the lung. Alright, so and the reason why vein they have to contain more oxygen is because they need to give it back to the heart to be able to go to the rest of the body. Yeah, okay, because our body accepts about 98% of the oxygen, whereas for the carbon dioxide, you only accept about 2%. Yeah. Okay, you also need to tell me what's aorta. Aorta is like the coronary artery and the anterior branch leading to the head and arms, as well as the, okay, this one, a posterior branch, this one you don't need to know. Alright, this one is only covered in G3. Yeah, okay, G3 pure. Yeah, okay, and the last one is, oh sorry, the next one is vena cava. Vena cava, VC. Okay, you need to know it carries blood from the rest of the body back to the heart, located on the right side of the heart. And because it is about the same as pulmonary artery, okay, it contains more carbon dioxide. Can you see a similarity here? Okay, because it has to be transported back to the lung, okay, for gaseous exchange. Alright, and the last one is the hepatic portal vein. It basically transports blood from the alimentary canal to the liver, and the blood from the liver is returned to the heart via the hepatic vein. Hepatic vein, okay, is vein, hepatic vein, very important. Okay, then the component of blood, you need to know that it has red blood cell, white blood cell, platelets, and plasma. Alright, so let me explain. Red blood cell is basically, it has no nucleus and a biconcave shape. That what we covered in chapter 1 just now, under cell structure and organization. Okay, that why you don't need to know. This is important. White blood cell, okay, it forms part of the immune system, and they are also known as leukocyte. It's okay if you don't know how to memorize it. Alright, but you also need to know it also undergoes phagocytosis when they engulf and digest foreign particles such as bacteria. That is phagocytosis. Okay, lymphocytosis is basically when they produce antibody that help phagocytes recognize and neutralize foreign particle. Okay, particle that's unknown. Okay, next one is platelet. Platelet is to stop bleeding by clotting the blood flow. Okay, and you need to know that it catalyzes soluble fibrinogen into insoluble fibrin thread. Okay, these are important as well. The last one is plasma. Plasma is when 55% of blood is plasma. The liquid part of the blood is 90% water, and you know it transports mainly RBC, WBC, and platelet. Okay, the function of blood you do not need to know. You just need to know that oxygen is for respiration, and you just need to know oxygen, hormone, and nutrient. So hormone is like the important chemical substances for various chemical activity. Nutrients such as glucose, amino acid, fatty acid, glycerol, and vitamin, you also need to know. Okay, also transport the following substances for the body cell: carbon dioxide, as well as urea, uric acid, creatinine, excess water. Okay, you just need to remember excess water. That's all. Alright. Okay, for the blood vessel, right, you need to know about the valve thing. Okay, what are valves? Okay, the lumen for artery is small. For vein, the lumen is big, is large. The capillary is very narrow. Okay, the thickness of wall, arteries is thick, veins is thin, capillary is only one cell thick, means it's very very small. Okay, the moment you damage that one cell, your capillary does not work, no gaseous exchange, will lead to a hole in the heart, which is a very important problem. Okay, the valves for the artery are absent. The valves for the veins are present, and the valve for the capillary is absent because there's no space. Okay, blood pressure for artery is high, vein and capillaries blood pressure are low. Right, so you need to know that valve in the vein prevent backflow of blood. Okay, so the heart, you need to know the structure. You need to know that it consists of the right, left atrial, and ventricle. So you need to know that the atrial is the upper chamber, ventricle has thick muscular wall. Okay, the left one has thicker walls than the right. Okay, because they need to pump blood to the rest of the body. And the right ventricle pumps blood at a lower pressure as the blood only needs to travel to the lungs. Okay, shorter distance from the heart. Alright, these valves, okay, between the right atrium and ventricle, which is here, right atrium and right ventricle is right here, this part here. You will see this small thing here. This is your tricuspid valve. Tricuspid valve. Okay, tricuspid valve, right, is when your heart is beating, is beating, you can hear the loud sound. Okay, the reason why you have the loud sound is because of the tricuspid valve opening and closing. Right, it opens and closes. Okay, and then between the left atrium and the left ventricle, which is here, this is your bicuspid valve. Okay, it only contains two flaps. That's why it's called bi. Bi means two. Okay, they are called. Okay, then the locator at the start of the aorta and the pulmonary artery, which is here. Okay, you can see this. You can see this small valve here. Okay, that is your semilunar valve. Alright, cardiac cycle you do not need to know. This one important. Coronary heart disease. What is the what does it mean? Okay, it occurs when the coronary arteries become blocked or narrowed, and the heart muscles will no longer be able to receive sufficient oxygen and nutrients. Okay, means basically coronary heart disease in Singapore, coronary heart disease is the #1 cause of death. Okay, so it's a very scary thing, and you do not want it to happen to any of us. Okay, so this is basically known as hole in the heart, because when they are not able to receive sufficient oxygen and nutrients, and when the arteries are blocked, right, the thing can expand. Okay, the artery wall can thicken. Okay, which causes the lumen to become narrower. It will increase the blood pressure, and therefore it will have a hole in the heart which mixes oxygenated and deoxygenated blood. You may have a heart attack. Okay, during heart attack, blood supply to part of the heart muscle is completely cut. Okay, because the coronary artery is completely blocked. When the affected part die, it can lead to heart failure. Alright, and the cause is basically because of atherosclerosis. Okay, basically it's the deposition of plaque which causes the lumen of artery to become narrower, and therefore it will increase in blood pressure. And there are some factors that comes into consideration due to factors that contribute to high blood pressure. High intake of cholesterol and saturated fats means if you have high intake of potato chip, eggs, especially egg yolk, egg white, and like potato chips, like such as potato chips and your eggs. Alright, it will lead to high intake of cholesterol and saturated fat. Okay, number 2 is stress, which is something I want to emphasize. Please do not be stressed over this exam. This exam is only to help you, okay, to be able to pull your marks out in case your chemistry does not do well. Okay, stress, if you are too stressed, it can lead to heart problem, because when you are stressed, right, the blood pressure will also increase, unfortunately. Alright, and the third factor, which is the most serious case, is smoking. I hope none of you watching this video are smoking right now. If you are, I suggest you quit, because if you are smoking, it causes your blood pressure to increase. You have a lot of black particle in your heart. It will lead to coronary heart disease. Okay, not lying. And there are some preventive measure to not have coronary heart disease. Firstly, have a healthy diet. Okay, take low cholesterol food and saturated fat food. Take less potato chip. Take less snacks. Take lesser eggs. Alright, these are the things that can contribute to high cholesterol and high in saturated fat. Number 2, no smoking. Say no to smoking, because there's a substance in there called nicotine, which is super addictive, which will increase your blood pressure. Okay. And number 3 is exercising. Lower your stress, strengthen the heart. Right. Please have a healthy diet, yeah. Okay. No more potato chips. Okay. Try not to eat too much, I say. Okay. I'm not asking you to quit completely. I'm just telling you that you eat too much, higher chance of coronary heart disease. Okay. Yeah, and that's all for the chapter. Nice. Okay. So we have covered everything from chapter 1 to 5. Yeah. So that's about it. Good luck and all the best. Good luck and all the best to you. Alright. For your end of year paper, this is the final stretch. Alright, please do not give up. Okay. As I always say multiple times, if you have a dream, you keep fighting. Okay, you keep fighting for your dream. Chase your dreams. Alright. Your dreams are not over. Alright. This subject is worth 50% of the combined sciences subject, which is a lot. So I advise you to make sure that you really put in the effort to memorize. Okay, because it's very, very important, especially for the one on the coronary heart disease. I suggest you at least take it down. Alright, I suggest you at least take down the coronary heart disease, as well as the factors and the preventive measure. Alright. Okay, so that's all from me. Thank you, and the next time I will see you will be after end of year exam for our post-exam activity. Yay. Alright, thank you, all the best, and goodbye.